hanging out on
Wow.
maybe tube worms or something.
Those are cool.
Oh, they're. I don't know what they're on. They might be on a little coral or something. Little black corals.
Can you bump a little closer?
What's that?
You need me to bump a little closer to you?
I
I guess we should figure out what the, so.
Yeah, another 10.
Yeah, roger.
If we're gonna move along this feature, yeah.
Is that the intent?
Or another
Science?
20.
And we can zoom out and move along. That was nice.
Do you want to move along this feature or head to 1F?
Let's move along at about, I don't know, 10, 20 meters and then head on
Roger.
up to 1F. How's that?
Sure, sounds good. I'll just go
Just
west.
take a quick look.
Yeah, 20
Bridge
west will work.
Tom suggests that these amphipods may be on
20 meters west. Thank you.
Thanks, Tom.
And we saw a little bit of this last night. I think just as Erica was getting off. We have Brittle Star, the paler.
Zoom out just a bit, Steve.
Yeah.
That's good, mate. See if I can swing it around here without killing it. Hey, who's that hiding in there? Good, do a full zoom there.
Coming in?
Yeah, Roger.
Could be
See
that
you.
boy.
There's a Tritonia under there as well, it looks like.
Mm-hmm.
Okay, Steve, you want to zoom out a bit for me again? Let's get the lasers in view there. Okay, zoom right in there. Hold on. Pilot failed. Okay, you can try a slow full zoom there.
Full zoom.
Beautiful. So move complete, is it?
Ship move is just now completed. I think Argus will still swing a bit west. It's giving you a bit of leash though.
I'll give it a few more seconds here and then we'll mosey along.
Sure.
So these are feather stars. I think this is Florimetra. They are suspension feeders, so they are picking out water, particles in the water column. And those little pinnules that cover the arms, those are used for feeding. And there are tiny tube feet on those pinnules, and they secrete mucus, and that aids in catching food particles as they're floating by, such as marine snow or little zooplankton. And if there's dense aggregations of these, it may indicate where food is abundant. So maybe this is a good place to live, this little condo area.
And they look very sedentary, but they can suddenly up and move and actually fly using all of their different appendages.
Yes, it's fun to see them swim.
The water.
Okay, thanks. Move on a little bit.
Okay. I can zoom out, please, Steve. We've got a hitchhiker.
Yeah, if we could just move along at this sort of perspective, that would be nice, if it's possible.
All right.
Erica, did you see guys' messages? We could follow this outcrop, but it will remain mixed, or go roughly east into the middle of the high backscatter.
Okay.
So right now the Argus is pretty stationary. We were just gonna check this out until the tether runs out. If you want to continue it, I could step more northwest, or we could then go east to 1F.
Let's continue it till the tether runs out, and then we'll head east to 1F.
Okay, sounds good.
That sound okay?
Works over here.
Nice to see this little community here.
Aletharians hanging out. I was at a little sponge down there under the thorny head.
Yeah, looks like a little sponge.
Yeah.
I do a laser zoom there.
That would be great.
Sure.
Zoom in there, Steve, to just keep the lasers in the shot. Yeah, perfect. Thanks.
Interesting thing to the left of it. Maybe is that a little tube worm sticking out with its?
And
Yeah,
to
like the
right.
feather worm.
Yeah.
You want to bring up the DSC on that engineer computer, just so we can see what the... I don't think we'll be able to see. She's got that.
Remote desktop.
Yeah, you'll have to re-log in.
Yeah, I can do that.
You get a couple
Never
of
mind.
frame
No,
grabs without this.
you won't be able to move the window unless you start another log file, which is painful.
You get a nice frame grab without those lasers. Just one or two.
I think we're doing that. We should have hopefully got some frame grabs by now.
With no
without
lasers.
the lasers.
No lasers?
Turn the lasers off.
All right, hold on. Let me get him centered up again.
I think, is that a stalk on that sponge? Or
There you go.
is that a
Hopefully we're stable there.
Kind of
It
looks
could
like,
be a stock.
yeah, could be a stalk under there.
Okay, so much for that. Never mind.
Okay.
Okay, thank you.
We have a suggestion from Science Chat that maybe we're seeing more nudibranch eggs.
Sorry, what was that?
The object below
Oh, I
is
was
actually
saying
a sponge.
That's a sponge.
You can
Yeah.
Maybe it was.
put the lasers on and zoom out just a bit. Yeah, maybe a bit more. Yeah, good.
Thanks, everybody. That was nice.
Is that a glass bottle
I
there?
have a hermit crab.
Yeah, it looks like
I
it.
sent that message in a bottle last time we were out here. I was wondering why I never got it.
And what did it say?
Secrets.
Here we have a hermit crab, a Tritonia nudibranch,
So feather star
some
really
human
going
debris.
crazy over there. Fighting the crab.
Yeah.
that clear anemone-like thing going around on the left, that's a corallimore. You don't need to go back to it, just wanted to point it out. Sometimes these ledges can serve as a little hideaway for rockfish, big and small.
And a shrimp.
It really makes sense that the more complex the structure of the habitat, the more opportunities for species diversity. Or potentially.
Yes. With all these snails around here, I might expect to see some of those more egg cases that are stacked. As long as there's a hard substrate.
Okay, I'll call in a move towards 1F,
Great, thank you.
bridge nav. 50 meters, 065. 50. Thanks. 065. Thank you.
So just a reminder that the two green lasers in the center of the screen, the 2 points are 10 centimeters apart.
It's like there's another ledge coming up.
Should have some time to look at it as we go.
Maybe this is the long promised cliff. Somebody told me there was a cliff on this deck.
So we're just passing a lobed ctenophore that was in the water column. I've seen a couple of those on this dive. That's a nice little ledge.
So in science chat, Guy is mentioning that what we've been seeing is softer layers of mud that erode beneath the harder sand layer. So I believe, Guy, you're talking about the creation of these ledges.
Yeah, if you lateral south, I think, along the ledge.
Oh, give me just a bit.
Yeah, I have to turn while I'm facing it because I'm getting pulled on my tail there.
Yeah. It's a nice little, some coral. Thought I
Yeah.
saw a sponge back there. There's this little habitat.
Yeah, some soft corals and hard bamboo coral there. Is that a paragorgian?
It is a little one, yes. Little nubby one. So I think Gary is in the science chat. If there are any corals he would like collected, please let us know.
Stop for a minute.
Sure. Bridge nav. Hold position.
We'll
Thank you.
come back the other way on our tether.
Yeah. Sounds good. South, east. Cool shot.
Guys
Like it's a
chiming
little
in.
spotty.
We're hearing from Guy that this is Rugos if you want to follow it for a transect.
Okay, well, let's do that. Are we able to do that, Rennie?
Sure, yeah, we've just stopped the ship. We'll see where Argus settles out, but I think we'll be good. It's hard to know the extent of this, so with Dan going
That's okay.
southeast, we can kind of start at that end and then actually come back up. Or we can start now and continue. It's up to you if we want to head northwest or southeast along it.
Let me look with the sonar and see which way looks more exciting.
Sure.
I can get it.
Okay, I'd like to make continuous headway towards 1F if we can.
So 1F is
whenever.
yeah, kind of behind us, and this ridge kind of follows the contour that we're on right now. So it is a bit northwest and southeast. So in order to follow it, we will have to be kind of running parallel to 1F.
Maybe we can just continue running it like you're doing.
Okay.
And
Porch light off, is it?
if it looks good.
You can kill that one. Thank you.
Might have seen a couple
Bring your
of
head to
spot
the west a
lobsters.
little more.
Roger.
Hard to return on this direction to the south.
Okay, so maybe we start here and we go south-southeast or
Sure.
south.
Yeah, south-southeast.
Great.
Okay, let's.
yeah.
You want this to be an official transect?
Yes, please.
Okay,
And we'll
so
just run it as long as we can
okay.
for
Roger.
150 meters.
So this is
If we run out, we run out.
T9B, if I'm not mistaken.
Yep, that's the start of transect.
Yes.
T9B.
T9B, 10-10-50. And which way do I need to move this thing? Argus is
Zoom in there on the coral, Steve, while we're getting set up.
looking east.
Yep, sounds good.
So, well,
That's good. Perfect. Thanks.
just take a stab at it and go 150.
That sounds about right, ain't it?
Yeah. Bridge nav. 50 meters, 150.
Erica, do you want a
Can you slow
picture
down
without
to
lasers?
0.3, because we'll be going with the wind
Roger
here.
that. Bridge nav. If we could slow speed to 0.3. Thank you.
Sorry, what was that in the background?
Oh, I was wondering if we wanted a glamour shot without lasers.
Oh yeah, kill the lasers, please.
Yep.
Really cool. A lot of stuff in the shot.
So these are primnoid corals.
Bummer we can't see our
Yeah.
DSC to see if it's framed up.
Tom Leidig say it's Peristanella. It's a genus of coral.
Might be worth stopping it and starting it. You got to, you know what to do there?
What was that? No, I know nothing about this.
You'd have to create a new folder on the desktop, _3. Because when you stop that and start it again, it's going to ask to erase all the files in that folder.
Oh, I see. Yeah.
So
I don't know if I want to mess with it right now while we're.
yeah, I can see in the background. We're getting some good shots.
Yeah.
Yeah, I'm happy with that. Perfect. That's all I need to see, actually.
Cool. Yeah, I think when we were up close, we actually got some good ones too. Okay, I think we can.
Okay, start a transect.
Yeah, slower at one.
You got to turn the lasers back on.
Yep.
And then what's our zoom setting?
Yeah, looks good around there. It looks like we're a little hot on the camera there, are we? Or is it just me?
I think we're zoomed in a bit.
I can come wider. Yeah, I want to get closer to the full wide for the transect.
Yes, please.
All right, coming wide.
Just a touch more, Steve.
Past that flare. Okay.
Right in there somewhere. Have to get the ROV closer. So yeah, when we're zoomed in, I tend to pull back. So. There, that's what we want. Some light on the scene.
Crispin Little points out the two stone crabs with the gray carapace and red legs.
You can leave it up there if you want. I can see around that silly dialog box. Just for a few minutes.
Box is annoying.
Yeah, so when you start it, you have to move the other dialog box because then it doesn't let you move that one. It's kind of retarded.
There's a mushroom coral that's with all its polyps out.
Right in the middle of the scene.
Yeah.
I'm gonna have to move a little faster, and he's still moving, is he, or is he gonna?
We're 0.3. We're still going. Yeah.
Roger.
Do you want me to hold here?
Yes, please. Yeah. Let me catch up.
Bridge nav. Hold position.
But another egg casing kind of hanging down there, shaped like a pea.
No, did I?
Yeah, I couldn't figure out what that was.
Where are you looking?
It's out of frame now.
Oh.
Come down 3 meters straight, please.
Yep.
So that pink anemone in the middle, I think that's a pom-pom anemone. Like Liponema, sometimes you see those rolling around the seafloor, but they are able to, well, maybe it was, I can't tell. They can adhere to a substrate or just roll.
You'll have to bring your head off. Auto head off, please.
Rolling back.
Sorry.
Auto head off.
Yeah, I let it get too far ahead of me.
Yep. So you just have to bring your head around.
Yeah, just till I get a little closer, then you can.
Yep.
When I'm forced to fight it, that blows the cliff out. Or was moving too slow. La-di-da there.
always amazing to see all of these different species, so from such different families, coming up with kind of similar strategies for foraging with the long branching arms for suspension feeding.
Yeah, I think it's so interesting that things like to climb up into the water column as high as they can to try to help grab that food. It seems like it would, you know, hit the ground eventually, but it does seem to be an advantage because a lot of the vertebrates crawl up on those crinoids and on the corals to try to get a little bit of extra altitude for feeding.
Well, the water, yeah, that's a squat lobster in the middle, isn't it? Just passing to the right. As water flows near hard surfaces, it tends to, instead of just fully making 90-degree contact, it flows along. So it makes sense to be up above a little bit. Looks like a Canthogorgia here in the middle.
The yellow one.
Might be able to bring your head back around now. I think I'm under you.
Yeah, I'm bringing it around right now.
See a little glow in the down camera there.
This looks really great.
Seeing more of those polychaete worms we saw earlier when we were zoomed all the way in, kind of feathery red circles.
Mm-hmm. Couple of soft corals. The pink one's called Jersemia, another deep sea sorrel. They're so beautiful.
A lot of sea cucumbers. Couple of single stalk coral here. What do you think those are?
The mushroom coral is right coming to center with all its polyps retracted.
Yeah.
Wow.
And that anemone is not Liponema. I will get a correct name on that.
So I think, Jake, you want to spin clockwise and look to the east. So it looks like we're coming around to east course overground.
That may be a Swiftia fan up in the right.
Oh wait, clockwise to the east?
I didn't see it.
I think you want to go clockwise to take your turn out.
Yeah.
Put one in on me, probably.
Roger.
Not sure.
So those big anemones are actually a Syrianthid anemone. All of us biology geeks out here.
Common name being the pom-pom, is that right?
No, it's a different family altogether. They sure do look similar.
Let's you hold that for a minute, see what happens.
Some more of the Jersemia, the soft branching coral.
Erica, those anemones deserve an outrageous common name because they're pretty spectacular.
I think it's just called well, if I'm right, it's a tube anemone, which sounds not so exciting.
Here we have a whelk with an egg tower.
Wow, it's sitting right on top of that egg tower.
Nice.
I was fishing out here one time and I lost my fishing line. Oh, there's where I left it.
How's it
I knew I'd find it again.
Suspended?
been quite a while.
The thing about that fishing line is you leave it somewhere, it'll still be there when you come back. What is this, what kind of sponge is this up in the right?
Gonna go 20 meters, 110.
Hold
I
on,
do
I'm
not
right
know.
underneath him.
Okay.
Some sort of
Science
plate
chat.
sponge.
What do we think about this sponge? The shrimp, a couple of different shrimp inside. Common spot for inhabitants of the sponge.
I don't quite have it worked out.
Yeah.
Am I still right under him? Yeah, pretty much.
Pretty much.
Tom suggests that might be a Costolastra sponge. Forgive this pronunciation.
Square up here and see what it looks like, right?
Yeah. Kind of
A
looking
couple more mushroom
east, huh?
coral.
Yeah, it is looking eastish, isn't it?
That one has its polyps out.
And
A couple of them.
Try 100. I gotta get
and
you over there.
another,
Yeah,
another
Roger.
sponge.
Try that. Bridge nav.
I don't mind Argus being on this side that he's on now. It's all right for me, if it's all right for you.
What do you mean by that?
This is just beautiful in here.
It's on the uphill side, but that's all right. It's not that steep.
30 meters, 100. Take it. Yeah, I don't mind it. Wherever he lands seems all right.
It's a black corals, aren't it?
Might be tucked back in there. Hard to say. There's a big.
It's a hagfish festival.
Ooh, is that crab eating a hagfish alive?
Or is it the other way around?
Yeah.
That crab looks a little lifeless.
Yeah.
It does.
This is a fantastic stretch.
It was till I dusted it.
The soft coral, and then there's another mushroom coral. It's all very exciting.
Come
This is
down 5, Jake.
incredible.
It really is incredible.
Let's test out
Don't
that
worry about a little
10-meter
dusting.
minimum
They've seen-
altitude on Argus.
Speaking of, can you
they've
bring that
seen
sub bottom up?
turbidites.
Yep. Or engineer up over there.
Randy doesn't
Another
trust our analog
Talon
altimeter.
minus the whelk.
Yeah, I never do.
Is that another egg stack?
Yep, exactly.
Yes.
Looks like a corn on the cob to me.
You can bring your head to the right a little. Looks like
Got a nice
he's
variety of
stepping, is he,
corals
Randy?
We're moving, yep.
Roger.
Take a bit.
I need to wait.
Yeah, I'll have to go at snail's pace.
Roger.
Is that a paragorgia there? Oh, a sea
That, I
plant
think it-
swimming. Up in the
Yeah,
top.
that coral I think is Cibogorgia. It is related to Paragorgia.
Okay.
Quite a variety. This is nice. I think Dan and Remy ordered this up. They're like, we need some excitement. And they made it happen.
I wish
Thanks,
it worked
guys.
that way.
Well, we put in our time with that quiet transect class.
Yeah.
Taylor Ann wanted something to record. She's like, give me
Yeah.
some work.
Looks like it's going to curve around to the south now.
Yeah, Roger. After this, I'll dial in the south.
Roger.
Try to time it so we keep moving.
Another whelk tower. And a mushroom coral. Just a beautiful look.
Problem with Argus on this side, it's sucking me into the cliff.
Yeah.
Getting a lot of good pictures and notes back here of all the things we're seeing. This is great.
And
Keeping you
that
busy.
happens.
So for those of you who have just joined us recently, we are conducting a transect right now on some rocky habitat. So we're trying to cover
Is that drift
about
kelp?
150 meters. Yes, kelp.
We're on transect though, aren't we?
We are.
Yep.
Yes.
I can
Okay,
mark it if you
possible, yes, that'd be really great to collect to see if any microplastics are on that
I'll mark it and see if
possible
we
vector.
can.
Yeah.
Because we have to come back this way anyway to get to F. So
Okay.
on the way back. Although
I don't know. We're going south. We were going east and then southeast. Isn't that our general direction?
yeah, no, we gotta come back up here to.
Oh, really?
Yeah, we've gone quite a bit.
Maybe there's another ledge. It looks like we can follow a different ledge on the way back.
Yeah.
Unless you want to come back and get that kelp, of course.
You'll have to Alfredo it into a bio box.
Yeah, I don't think I've ever got kelp in the bio box before.
If possible, it'd be cool if we could slurp it so
That
that
would
way
be-
it's contained.
You would have it more likely to have it when we get on the surface that way.
We're likely to clog the hose too. Unless we could probably break a piece off of it. Because it was pretty long.
Is that a swift
Yeah,
bead simplex near
I've
the
slurped it before.
thorny head?
I don't know
I
if
don't-
it's slurped
oh,
a
yes,
piece
I
that
think
long.
so.
It usually tears. Looks like you got to look to the south a little, Jake.
Bridge nav. Uh, 30 meters south. So we have gone approximately 75 meters along this transect.
There's another feature coming up on this bearing in about 20 meters.
Yeah.
How far are we? We're supposed to do 100 meters or 150?
150 was the aim.
Yeah, the aim. If we get to 120, that could be fine because it's pretty dynamic.
Sure.
So while we're collecting this transect video, we won't stop if something, if we pass something of interest, like the kelp that we just passed. We may flag it to be able to come back if possible, but we are really trying to keep a continuous speed and continuous altitude off the bottom, collecting this video that will then be post-processed after the cruise by a number of different specialists, at least a specialist looking at it a number of times, each time for a different suite of species. So coral, sponges, fish. And on this stretch, we've really seen a great diversity of coral.
Guy was noticing that some of the bedrock he saw had broken off and slid down slope a little bit.
Yeah, I missed that. It's great to be able to have a variety of geologists and biologists on our science chat that can pipe in and provide advice where needed. Guy will be looking at these transects later and ground truthing some of the multi-beam and other maps we have.
I haven't picked up any
I'm
rocks
wondering
yet
if-
on this cruise. What's that? I got picked one up, but you guys wouldn't let me put it in the bio box. Too big.
Another mushroom coral. Oh, we didn't get that rock last night?
I don't know if there was another watch that picked up a rock.
We
There's
didn't
some-
keep the one that we picked up.
Do we have some rocks?
I don't know.
We picked up one sample number 2 last night.
Oh, yeah.
It's all a bit of a blur though, isn't it?
Must have been the 4 to 8 shift got all the fun stuff, huh?
Oh, right when we handed over to them? Yeah.
Yeah, and then they filled up the sample box, starboard sample box before the H12
Yeah, what's the
even got
status
here.
on the, do we have any sample boxes open?
Yeah,
Out
we
of
still
curiosity.
have 2 large boxes open in the forward bio box. We haven't used those at all. And then in the starboard bio box, it is full, but we could possibly pair some sponges with corals. Just making sure we're not mixing any of those corals and corals together though. But there's room. But we do have 2
Or
completely
sponges
empty.
and sponges.
Yeah, we do have 2 big forward bio boxes that are empty. And then all the slurps are empty. And then, yeah, if we need to pair up, we can pair some things up in the starboard bio box.
What do you think? Another move south?
Yeah,
Not really moved south, has it?
yeah.
Not really seeing much on the sonar. It might be the edge of it.
Just looks like there's
How
another
far have we
feature here.
traveled?
We have traveled approximately 85 to 90 meters with the squiggle. Yeah, 90,
Okay.
90 meters.
I think there's another
If
little
we can get to
feature right here.
120.
Okay, sure.
It might be a mixed habitat, but there was a lot of rocky there, so.
There's the ledge kind of petered out for a second, but I think it's here in front. I can't tell for sure. Yeah, little micro one.
I think we'll approximately accomplish our transect without another move.
How many meters have we covered so far?
Let's see. I'll try to do a more accurate measurement here. I'd say about 100.
Okay, let's try and go to 120 unless we run out of this ledge, and then we'll just stop. But if we can make it to 120.
Yeah, it looks like it goes on for another 20-ish. Close. According to our sonar.
20 meters, 135.
Sure.
Ridge nav. Two 0 meters, 135.
Guys suggesting
Guys
to
suggest
go to those 2 bright pixels.
Sorry. Say again?
He takes it back.
Guy was
Never mind. Comment removed.
Takes it back.
Okay.
Follow the ledge.
Follow the ledgy, he says, yeah. Thanks for catching that, though.
the affectionate term for ledge.
Yeah, we were guessing the vessel move there based on what we're seeing in our sonar to try and keep going along the ledge.
So the ROV heading in a southwest direction, is that our direction at the moment?
Southeast is the way that our course is going. And
Okay,
we've been facing roughly southwest as we go. crabbing along, if you will.
Yeah, so as Guy points out, we have changed substrate here. This is definitely mixed habitat, even going into soft here.
Sure, so let me know if you want to continue on or if we want to end the transect.
Yeah, we're probably at 100 meters by now.
Definitely,
Or more.
Yeah, over 100 for sure.
Yeah, let's end it.
Okay.
Is that it? And the transect?
Yeah.
Roger.
Thanks, guys.
Sure. See, remember how off we were from the DVL, and now we're right back on.
Yeah.
It's that USBL funniness. So I kind of, I don't know, the resetting. I'm making sure I'm not over resetting.
Needs to do it every minute.
Yeah.
Automatic.
If I trusted the
So I
USBL
think
more, I would. Sorry, continue.
we had discussed if we had some time to head back to that marking point of the kelp.
Yep.
And that's going to head us back to 1F. Is that right?
Yeah, it's directly on the way.
Okay, great. Make Taylor Ann happy. Let me see if there's any microplastic. Hey, is there some right in front of us?
Yeah, we'll see
Or
if
is that
it's
ledge? Oh, sorry, that's a ledge.
Yeah, see
Okay.
if kelp is a vector for microplastics. So part of my master's thesis is studying microplastics in the deep sea, and then the other part is studying microplastics in kelp forest ecosystems to see if the thick densities of kelp that change the physical environment and slow down the flow in kelp forests actually, if they could potentially cause
I didn't know.
for kelp
Bridge
forests to
nav.
be,
Hold
like,
on, Rennie, let me
I
get
guess
out. Oh, you
hotspots
can
for
move him.
microplastics.
Sorry, go ahead.
30 meters, 315. And we're roughly going to that cross that I dropped.
Yeah, roger.
But we will.
Boot scoot to the next sonar target here.
So Taylor Ann, is it thought that the microplastics adhere to the kelp in the shallow water, or
we've
they
obviously
collect them
been
on
here.
their way down?
So nobody's really
Wanna
looked
spin
at
around?
the species of kelp that I'm interested in, the
Kelp?
large kelp,
Yeah,
Macrocystis porifera. But there have been in-lab studies of smaller algae
just gonna
that have been able to retain microplastics on their
fly
surfaces.
rapidly to get through the dust storm here.
And then organisms have ingested it. So like organisms such as snails that feed on algae have been seen to ingest microplastics from the surface of the algae and then reintroduce that into the environment again through feces. So part of my project is seeing if giant kelp, Macrocystis porifera, a larger kelp with a more textured surface and mucousy surface, if it could adhere, or microplastics could adhere to the surface. And so potentially kelp forests could be hotspots for microplastics, or even potentially act as filters, absorbing or causing all the microplastics to kind of stay in one place. And then if that was the case, we could probably see microplastics being carried along drift kelp into other ecosystems, such as the deep sea. So we'll find out. Nobody's looked at it really, so we'll see.
All right, interesting. We have been doing some microplastics collections above Davidson Seamount, as well as in the sediments, I believe.
Yeah.
Or at least in the water at depth, perhaps in the water at depth.
Yeah, trying to see the different concentrations and types of microplastics associated with different water mass properties. So at different depths, that'll be something that we're looking at with some Niskin samples.
It's a nice rock there. So fit on the
and
front porch.
Gathering some holothurians
That would fit in A.
and suspension feeders to see if they're ingesting microplastics as well down here.
I think
Interesting.
there was someone out, and it may have been on the Monterey Bay cruise last year. Can't recall his name, unfortunately. Looking at PFOE in the sediment as well.
Yeah, Dane Harden.
That's it.
Yeah.
Dane.
He was on board. Yeah.
Nice guy.
PFOE. What's
Yeah.
what's that stand for?
A long chemical name. Polyfluoro something.
Yeah, he's looking at persistent organic pollutants, right?
Yeah.
Yes, correct.
Which, yeah, microplastics have the potential to carry those along with them too, other like toxic chemicals on their surfaces, which is why we're really concerned about them finding their way into the food web. If we're introducing plastics into the food web and organisms are feeding on them, it's making its way up from small organisms like copepods all the way up to fish and potentially onto our dinner plates. And we want to know what we're eating, and we don't want to be eating plastics and harmful chemicals. So it's something that we're trying to monitor and kind of get a grip on of where they exist, and if they are there, in what concentrations and what types, and what implications does that have on deep sea organisms, other pelagic organisms, and humans.
It's great that you're working on that.
Yeah.
Zoom in there for a minute, Steve, while we're waiting for the boat.
If you have a kelp sample from this cruise or other sampling, how do you process it to kind of find those microplastics? And it kind of sounds like our slime and hagfish discussion, I'm imagining, if you have old kelp with things stuck to it.
Yeah, so my plan with the kelp would be to try to collect it in an isolated container, so potentially a slurp jar if that's possible. So that way I can actually filter the water that was collected along with it as well. And then I would also rinse the surface of the kelp and save that water and filter it. And then eventually chemically digest the organic material from the kelp. And once that's broken down, I would just filter that as well. And then I'll use this fancy machine called a micro FTIR that uses infrared technology to tell me what types of plastics we're seeing, if they're plastics at all. Because there's a whole catalog in that machine that has the different plastic types available, and it'll be able to tell me what we're looking at.
Move complete, is it?
Move complete. Yeah, I think you might have the leash soon. Looks like you
Okay,
got some.
you're gonna zoom out, please, thanks.
So that was a Swiftia coral we were zoomed in on.
So this
Small
kelp
red
was
coral
on
with the
this
dark
ledge
skeleton.
somewhere.
It was.
Okay, Taylor Ann, do you remember what it looked like, what the environment looked like to pick it out?
Having fun back here. This is great. So different from last night where we were just seeing sea pens. So this is sea pens are cool too, though.
There it is, I think.
Yep, it looks like it.
See, I'll bump you a bit closer.
Roger.
Yeah. Bridge now.
I'm
15
amazed
meters,
you found it.
300. Thank you.
Okay,
So
Jake,
I'll
you want
correct
to
myself.
turn on the camera that's looking at the sample jars, and you can also turn on the porch light.
Roger. Turn on porch light.
And when you get a chance after all that, you can look at the manipulator with the bubble camera.
Yep.
I'll correct myself again. That is Liponema, the pom-pom anemone. I promise not to go back again to the other ID.
You want to zoom in there, Steve, and get a close-up on the kelp before we sample it? Any particular place you want to look at it, Taylor Ann?
Yeah, it looks great.
Oh, flutist has been here.
That's full zoom there.
Roger.
This looks like a different species from Macrocystis, but still want to look at this. This would be awesome.
So will this be sample number 8?
Yes.
Not sure where we are.
Number 8. Yeah.
You want to show me the suction sampler or the nozzle?
Yep. Down.
Cheers.
Yeah, this looks like a blade of Nerio, so bulk out.
Yeah.
When you zoom in like this, you see so much more life, little critters in here. See a little tube anemone or tube worms, some caprellids, little shrimp, smaller polyps of something.
All right, where's the bottom of that thing?
Let's flush first.
Hydroids.
I know.
Brittle star,
Oh.
swift to you to the right.
Can you look back to your right a little? I might have to set
Waving
it
on
down
the
and
right.
get another grab. Right,
Oh,
other way.
right.
What are you holding on to?
I don't know.
That was funny.
Which jar is this going to go into?
If we haven't done any, I'll put it in jar number 1.
Okay. Yeah, we haven't done any yet, so that works.
The coral cutters are so like,
like so snaggletooth.
yeah, got like
All right, flush.
alligators are all trying to grab something.
Okay, you want to zoom out a little for us, Steve? That's good. Okay, we need to flush now.
Yes.
Turn her on 100%. Somebody really needs to paint that nozzle.
Then you already you painted the other one, and then that one had a hole in it, right?
All right, flush.
Yeah, let it run for a minute to spin in the mud and stuff. Still at 100%, are we?
Yep.
Roger. You'll see the big dust cloud clear out when it.
Taylor Ann, have you been able to get many kelp samples from these deep sea waters?
Okay, if you
I
stop
haven't
it there,
collected
then
any before,
go,
but I've seen them.
forward to jar 1.
So on my last cruise experience last year, I did
reverse
see
to jar
some
1 in this case.
some drift kelp last year. But due to COVID, I haven't been able to start my field research
Crank it
yet. So
on
this is my
again.
first
See if it comes through.
time out, getting to do real science, and I'm excited. And then I start my other scuba diving portion of my project in March to
Go for
survey the kelp forests.
yeah, go for 50% there.